All right, everyone. So let's jump into passage 1 here. So this is passage 1 from the Bio and Biochem section from the MCAT official prep sample test. All right. So here, this is just the first passage, and it's going to have 5 questions afterwards, which we will answer. But I really wanted to just show you guys how to use the strategy that we talked about earlier to digest and break down this passage into a nice pictorial representation, so we can always refer to a very easy to understand diagram for answering our questions, and hopefully that'll let us answer our questions a lot quicker, a lot more effectively, meaning getting more questions right, and overall helping us score a lot better. All right, so let's jump into the passage. So first things first, we have the first paragraph, which is always going to be some sort of introduction to the concept or the topic that we're talking about. So usually there's nothing that we need to take note of particularly there, so we don't need to really incorporate that as part of our pictorial representation. So first, we're talking about the proper biomolecular trafficking, including protein packaging by the Golgi apparatus, so an organelle we're already familiar with, and that's essential to eukaryotic cells. All right. Next, therapeutic agents that disrupt the function of the Golgi apparatus can reduce cell viability and be essentially effective treatment options for cancers. And that's something that we're actually going to talk about later on. And so usually the introductory paragraph sets us up for the whole rest of the passage. Now, the second paragraph is actually where we start jumping into our pictorial representation and actually drawing it out. With a quick glance at the paragraph, we can clearly see that we already have a lot of different shorthands and acronyms written out for us. And so that kind of tells us that there's a lot of key players here. There's going to be a lot of moving parts. The best way to tackle this is using a strategy that makes it all organized. And so the first tip that I usually tell is to have everything in a nice chronological order, right? So you're going to have essentially different scenes. And so we're trying to go ahead and draw out what's going on in the passage from beginning to end in an easy to understand connecting format, so we can kind of make sense of the whole system and really understand the big picture, because that's really what this is all about, getting the big picture. There's no need to get boggled down by all this information, because some of it's going to be useful, some of it won't be useful at all. And so really just getting the key pieces of information and separating the relevant information from the irrelevant, like we talked about before, is the key thing that you're being tested on in this section. So let's get started. The second paragraph starts by talking about ADP, or ribosylation factor 1, ARF1. So right there, I'm given a key player. I can tell because there's my nice little shorthand, and it really, right off the bat, just says right afterwards, plays an essential role in vesicle formation. And so we can already tell from the first paragraph, we're talking about protein packaging by the Golgi apparatus. And so we can clearly see that this is going to be playing a key role in this whole process. And so the first thing is, I'm going to number my first scene. My first scene is going to have this ARF1 guy. All right. And so he's going to play an essential role there, and he's responsible for the recruitment of cytosolic coat protein complexes, COPs. Okay, so first things first, focus on key words, right? So we got recruitment. So it's going to lead to the recruitment. So I'm going to go ahead and quickly write that out. Recruits COPs, one major function. Next, here we see that it also is going to lead to the subsequent retrograde transport from the Golgi apparatus. So retrograde transport is the second thing afterwards that takes place. So I'm not going to worry about what retrograde transport is right now. I'm going to focus on getting all this information down in a nice, easy to understand format. And then if I really do need to recall what retrograde transport is later on when I'm answering the questions, then yes, I'm going to jump in and then do that. But until then, there's no need for us to really think too much about any of the actual concepts that are being presented in this passage. Our goal is just to focus on the strategy, save as much brain power as we can, translate this into a nice, easy to understand format, and then we can jump into the questions and then use our brain power where it's actually useful. All right, so the next sentence is ARF1 is activated by guanine nucleotide exchange factors. All right, so we got keyword activated right there. All right, and so how am I going to represent that? So we got GEF factors. An arrow is going to directly indicate to me that that is activating that molecule. But how does it do it? Replace guanosine diphosphate, GDP, right here, with guanosine triphosphate, GTP. Okay, so now I need to represent what's going on. It's going to replace that. Now it's basically going to be converted into that. Okay, So that's what's going on. This little dotted arrow tells me what's happening after the activation process, okay? Next sentence. Here it says, upon GTP exchange, ARF1 undergoes a conformational change, releases this N-terminus of the polypeptide chain from a structural groove in the protein, and initiates localization to phospholipid bilayers. A very wordy sentence. Usually when it's something that's pretty wordy, it means that it's not that important. But I'm still going to represent this because I don't know if I might need this later on in a very quick and simple format. And so basically what's going to happen next, it talks about releasing this N-terminus of the polypeptide chain and initiates localization to phospholipid bilayers. And so I'm just going to quickly draw a little mini phospholipid bilayer. And that tells me what I need to know from that sentence. All right, next we jump in to the next sentence, and it says, once associated with a bilayer, ARF1 further facilitates vesicle formation by the recruitment, okay, so we've got another keyword, of the heterotetrameric dimer of dimers codomer protein complex. Okay, and then here it says COP, and so that makes me think of the COPs that we talked about right there in the beginning. So it recruits COPs. These are probably what those molecules are. And then we have subunits are represented by beta, delta, gamma, and then another Greek letter that I don't know. So, all right, that's the next thing that we see here. And so I'm going to go ahead and draw that out in my next scene. So we have ARF1, GTP. And so what does it do? It recruits those guys. Okay, so I'm going to draw some kind of arrow to just show that there's a relationship between these guys right there. COP, beta, delta, gamma, and that. So we got 1, 2, 3, 4. All right. ARF1 GTPase activating protein. So we got GAP right here. It's going to catalyze the conversion of ARF1 bound GTP to GDP and inorganic phosphate, thereby converting the protein to the inactive form. Okay, so that's an important piece of information. Catalyzing, another keyword right there. So what's going to happen? We see that we're going to have this guy, GAP, come on in, convert this into ARF1 with the GDP. Well, they're connected, obviously. GDP plus inorganic phosphate, and this is actually in the inactive form. That's an important piece of information for me. Last, gap activity is increased by ARF1 binding to this whole... Okay, so this guy, ARF1, binding to this whole molecule here, the COPS, is going to lead to the increase in the activity of this. So I'm just going to quickly represent that. I'm going to draw an arrow connecting these guys to show that this is binding with it, and that's going to go ahead and lead to increased activity right here. I'm going to use 3 up arrows next to the gap to show me that that's what that results in. Okay, so as you can clearly see here, this is very rough of a schematic. It's obviously not going to be exact. It's not going to be beautiful, but it gets the job done, as we're going to see shortly. All right, so the third paragraph is talking about Berfeldin A. BFA, a lactone compound isolated from fungi, has been shown to inhibit ARF1-driven vesicle formation, resulting in reversible disruption of the Golgi apparatus and tumor remission in vivo. Okay, a lot of good stuff here. So what are we seeing? Basically, first things first, keyword inhibit. So this whole process we talked about right here, scene 1, scene 2, BFA has the ability to inhibit that. So I am going to represent that in a third scene. BFA, I'm just going to point an arrow up, essentially referring to, draw that better, this whole process. I'm just going to say X, because it can inhibit this. And it's going to lead to tumor remission. And that makes sense. So this actually connects with what we talked about right here in the, first paragraph. And so here we talked about how therapeutic agents that disrupt the function of the Golgi apparatus, effective treatments for carcinoma, exact same thing going on here. BFA inhibits this whole process. It's going to go ahead and lead to tumor remission, so the tumor is not going to be in its active form anymore. Very good for our patients. And so that's exactly what we're looking at. We see that we're basically elaborating on what was talked about in the introductory paragraph, so very good. Second sentence in that paragraph, because of its low bioavailability, BFA is not a suitable candidate for pharmaceutical deployment. However, it has led to the identification of AMF26 as a promising drug candidate. Okay, another key piece of information. I'm given another molecule that's going to be doing the same thing, basically. I'm going to do an equal sign, AMF26. And then last paragraph right here. AMF26 is predicted to bind to a protein-protein contact interface of ARF1, preventing GTP exchange by GEF and disrupting ARF1 membrane localization in the initial critical step of COP1 recruitment and vesicle formation. Okay, clearly that's the most important sentence in this paragraph, and it's telling us how AMF 26 will actually inhibit this process. And I'm going to go ahead and include that as part of my initial scene, just so I can see where it fits into the whole picture. AMF 26 is actually going to, as we see right here, bind preventing GTP exchange by GEF. And this is where my GEF is in the first scene. And so that's why I'm drawing it right there. And I'm going to put an X right there because it's disrupting that whole process. And it talks about how it's disrupting membrane localization in the initial critical step of COP1 recruitment and vesicle formation. Okay. And then last sentence, in clinical settings, oral administration of AMF 26 has led to remission of breast cancer, xenografts, and mice model systems. So that's basically confirming for us that AMF 26 actually does work like BFA. And so that's why it's, as in their own words, promising drug candidate. And there we go. We have our schematic right there. So now we see that we've created essentially 3 scenes. We've got scene 1, scene 2, scene 3 right here. And that is telling us everything that is going on in the system. And now I can jump into my questions. And so why don't we go ahead and do that? All right. So question 1. Hydrolysis of the gamma phosphate of GTP bound to ARF1 results in. All right. So hydrolysis is something that we should be familiar with from biochem. It's basically talking about, well, lysis, the breaking or the cleaving of a molecule. And so here, that's exactly what's going on. Hydrolysis of the gamma phosphate, so a phosphate on the GTP. So GTP is guanosine triphosphate. We have 3 phosphates. So what happens when you remove one of them? It gets converted into GDP, guanosine diphosphate, right? And so if we have ARF, that's what we're talking about, ARF1 bound to this, what's going to happen? Denaturation, activation, inactivation, or membrane association. Well, let's go back. And that reminds me, the word activation in answer choice C reminds me of writing inactive right here. Why did I write that? To talk about how ARF1 bound to GDP will be in its inactive form. And so here, that's exactly what we have. ARF1 bound to GDP because of the hydrolysis of this phosphate group on the GTP. And that is actually going to lead to inactivation. And so that is going to be my right answer. All right. So question 2. The ARF1 activating molecule, GTP, is most closely related to which family of biomolecules? So this is actually not really relevant to the passage. What do I mean by that? This is a standalone question that has nothing to do with the passage. And yes, you may encounter questions like these as well. So these questions tend to be solely based on either simply making a small connection or from your previous content review. And so here we're just talking about the molecule GTP and which, essentially these are all classes of macromolecules, so which macromolecule class does it belong to? So GTP, as we already know, and as they already talked about in the passage, right here when they spelled it out, guanosine. So guanosine triphosphate is going to remind us of nucleotides, essentially, because if we think about it, we have A, T, C, and G. It should remind us of that G right there. And so this is obviously going to belong to that family of biomolecules, nucleotides. All right, next question, 3. So here, GAP belongs to what class of enzymes? And so now we need to be reminded of what GAP does. And so I jump to my schematic. Now this is an excellent use of my schematic. And so here, I jump in here, and then I see, okay, in my second scene, I have GAP, and what does GAP do? It converts ARF1 bound to GTP and breaks it down to ARF1 bound to GDP plus PI. So it removes the phosphate groups. And so that makes me think, okay, phosphatases, kinases, those are usually the enzymes that work with these phosphate groups. And so I'm going to jump in and see what my answer choices are. So here I got transferase, that moves a group from one place to another, so that shouldn't be it. Phosphatase, okay, that's one I was considering. Kinase, another one I was considering. And an isomerase, that converts from one isomer to the other. That's definitely not going to be it. And so now, obviously, I'm left with the 2 choices that I had predicted. What does a kinase do? Kinase is the one that'll add a phosphate group. Phosphatase is the one that'll remove it. And so that's why that's just something that we need to know from our content review, and that's going to be B. Number 4, proteins that are encapsulated in ARF1 COP-derived vesicles are bound for the. Okay, so basically, we talked about this whole process and this whole system. And what is this whole system trying to do? It's essentially allowing for the formation of vesicles, right? But here this question is asking, what's going to happen to those proteins? Where are they going to go? Well, let's just jump to our schematic, and if we do, we can see that in our first scene we had that kind of information already there. This is the introductory information, and here we wrote down that ARF1 bound to GDP, ARF1, this system is going to do two things. One, recruit COPS, which is happening right here, and second stage, which we didn't delve into too many details, but was mentioned in the second paragraph, RT, retrograde transport. And so now we do actually need to think about this concept. And what goes on in retrograde transport? Well, retrograde transport is going to be the reverse, so whatever the normal mechanism of transport is, hence the word retrograde. So what is the normal route of transport? Well, usually we have the endoplasmic reticulum, in particular the rough ER, is where we have ribosomes, which will allow for the formation of proteins for us. And then from the ER, usually we make our way to the Golgi apparatus for packaging and also signaling modifications, like adding another carbohydrate group, another signaling molecule, and then we send out our proteins for different locations throughout the cell. And so here, what's going on though is we have retrograde transport. All right, so that's the key word here. So that means retrograde transport, and then if we quickly do look back at the passage to remember, retrograde transport from the Golgi apparatus. And so it's going to go in reverse. So when we're at the Golgi apparatus, going in reverse takes us to the endoplasmic reticulum, and that's why the ER is going to be our answer. Now, a common pitfall is to look at this part right here and think that those proteins are going to be directed towards the cell membrane, but we do need to remember that that was referring to specifically a different polypeptide chain, that it's not referring to our protein products that are going to be in these vesicles and the vesicles that we're essentially trying to form as part of this whole process. All right, so question number 5 is asking us, based on the mode of action described for BFA in the passage, the drug would be most effective against. And so we have 4 answer options here: eukarya, viruses, bacteria, and archaea. So let's remind ourselves what BFA does by looking at our schematic. And here we see in scene 3, we talked about how BFA is going to inhibit this whole process that we detailed from scene 1 to 2. And so what is that process? It's the process of vesicle formation, and it's essentially talking about how this is going to happen through the Golgi apparatus, and specifically BFA is going to disrupt that process. And so let's see if we can use that information to answer the question. So the drug would be most effective against, here we have a eukaryote and the rest are all prokaryotes. And so we can kind of use that to guide our direction in answering the question. And what's the key difference between eukaryotes and prokaryotes? Well, first things first, eukaryotes are going to actually have Golgi apparatuses, whereas these other guys won't. That's the key difference. And so that means BFA won't be effective against viruses, bacteria, or archaea, but it can definitely be effective against eukarya for that reason. And that's why BFA is going to be most effective against eukarya, because they have Golgi apparatuses, and that's exactly the process inside of the Golgi apparatuses where we have this ARF1 molecule allowing for vesicle formation to take place. That is the process that's being disrupted by BFA. So that's exactly why BFA will be most effective against eukaryotes. All right, and so that's basically how you implement the application of a pictorial representation, and I'll use that to help answer your questions. And so here we saw how we were able to do that. In question 1, we were able to defer to scene 2. In question 3, we were able to defer to scene 2 again to see what gap was doing. In question 4, we were able to defer to scene 1 and take a little quick look at this quick piece of information that we wrote down here. And in scene 5, we were able to use question 5, we were able to use scene 3 to remind ourselves what BFA actually does to help us answer the question and see the discerning difference between the answer options. And then question 2 was just a standalone question, which really had no need for us to even look at the passage. And so the big takeaway here is that by charting everything, making these connections, simplifying the system at hand in these Biochem Bio passages, you'll really be able to jump at the relevant information, separate that out, and use that to help answer your questions in a lot quicker and more effective manner. All right, well, hope that was helpful, guys.